Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is December 5th, 2024, and we're talking to Jason Chen, who's professor in the departments of molecular medicine and ophthalmology, and the Joe R. and Teresa Lozano Long Chair in Retinal Signaling at our sister medical school here in San Antonio, UT Health, I guess soon to be called UTSA.
Yeah, I'm fine with that.
And his laboratory studies the molecular biology and function of G protein signaling in general, but specifically in photoreceptors and some other cells in the retina.
Mm-hmm.
So welcome, Jason.
Hi, thank you for having me.
Also with us today is Erika Tatiana Camacho, our local retina expert. Hi, Tatiana.
Hi, Charlie.
And I'm Charlie Wilson. So Jason, I think most of our listeners know something about G proteins.
Yeah.
Maybe not as much as they wish they knew. And there are many different variants of G proteins and G protein signaling used in synaptic transmission and lots of other ways in the nervous system and outside the nervous system. And I wonder if you could start by just giving us some, maybe G protein for dummies. We just sort of start at the beginning of G protein, and specifically with emphasis on how it works in phototransduction, because phototransduction is something very special. We think of it as super fast. We think we see the world in real time. I guess we don't quite, but almost. And we think of G protein as being slow. If I just ask somebody what they know about GABA B kinetics, they'll tell me, I don't know how slow, but I know it's slow.
Mm-hmm.
And so, but vision is fast.
Vision is fast.
So maybe vision is a great example for us to start with. So maybe you could...
And vision is the sensation you don't appreciate daily until you begin to lose it. So vision is actually a very good point to study G protein signal transduction for a reason that you can get real-time kinetic information. And vision is also a system where the stimuli can be very carefully calibrated and applied. Okay, so this is a big advantage of using vision. Of course, studying vision, you study photoreceptor first, because that's the cell that transduces light into a neuronal signal. So I don't know I qualify to give a general introduction about G protein signaling, but I hang out in Caltech for 5 years in my career, some 20-something years ago. And my professor, Mel Simon, he was actually given the credit of actually help us understand how important G protein might be for mammalian biology. So Mel's major contribution was actually in that old days when we still try to clone our stuff by degenerative cloning. So if you already know, you will know what this term means. That Mel figured out there are 16 G protein alpha genes, 5 beta, 12 gamma, and G protein being a heterotrimer, alpha by itself as a signaling moiety, beta gamma always exists as a dimer, 16 times 5 times 12. That is enough permutations to cover a lot of biology.
Help us understand why there's so many different.
Right. So then 1994 Nobel Prize was given to Martin Rabel and Alfred Gilman. Gilman is a Texas boy in Dallas. And then, they get Nobel Prize because this thing is so important. And within the two group, right, and a bunch of their scientific descendants, biochemistry of G protein is well mapped out. We know what kind of combination of alpha with beta gamma will, what kind of enzyme kinetic activity. So the challenge for G protein field has been, what G protein being used in what physiological context to do what, to regulate whom, and how long, and how efficient that regulation has been, that kind of stuff. So with that, so the field now into a challenging era to put physiological relevance of a lot of the in vitro reductionist finding of what G protein can do. So it is a challenge, but doing it in photoreceptor have the advantage of using genetic approach without having to turn yourself into a developmental biologist in most cases. And then anything that you made, the worst outcome may be just that your animal is blind, which in the vision field is not necessarily a bad thing. Because nowadays the NEI has this human population average lifespan being expected to be more than 80, right? And then there are the dreadful diseases like age-related macular degeneration, and the prevalence is pretty high. And if you're over age 80, 1 in 3 will have some form of macular degeneration. NEI is not prepared to answer a lot of the clinical challenges when it comes to taking care of those patients and what we can do as a community, vision research community, to help those patients, stuff like that. Yeah. So
can we kind of walk through the sequence of events that happens from the time a photon gets absorbed, the molecular sequence of events?
Of course. So in the rod and cone photoreceptor, allow me to just dwell on rod and use that. Then I jump to explain cone a little bit. So the two photoreceptor endow us with an input detection system from single photon detection in rod to like 10 order of magnitude higher intensity in high noon when you stare at the sun. Okay. So our system is able to respond. And then the rod photoreceptor responsible probably for the first 3 orders of magnitude of light intensity, and cone take care of the rest. Okay. Rod photoreceptors sense light by rhodopsin, which is the visual pigment that get hit by a photon. Okay. And the photon will photoisomerize a chromophore we call 11-cis retinal aldehyde. And that mysteriously reaction will isomerize the 11-cis retinal into all-trans retinal. And then that will cause protein conformation change. Within a few milliseconds, the rhodopsin molecule will rearrange the 7 transmembrane helices to a point that it will adopt conformation able to open transducing alpha subunit, DNA binding pocket. And then, GTP binding pocket, not DNA binding pocket. And then you will facilitate the exchange of GTP for GDP. When T-alpha, transducing alpha, binds the GTP, it goes through conformation change, and then it will dissociate from the beta-gamma moiety. And G-alpha, which is a signaling molecule, when it comes to phototransduction, the G-alpha in the GTP bound form binds to the PDE phosphodiesterase 6 gamma subunit. Okay. And gamma is an inhibitory subunit. And you will let the PDE alpha beta subunit unchecked. And PDE alpha beta subunit is a perfect enzyme. And the catalytic activity is equal to the diffusion coefficient of the cyclic GMP inside the cell. That means that when the two meet together, cyclic GMP get hydrolyzed immediately. So this kind of perfect arrangement, and then these two very fast cyclic GMP concentration reduction to a point, cyclic GMP stay bound to the cyclic GMP dependent channel, okay, lost the ligand, and the channel closed. And when the channel closed, and then the cation entry carry 85% by sodium, 15% by calcium, close. And then the system go into hyperpolarization.
So how long does that take?
Yeah, it depends. In mammals like us, right, 37 degrees Celsius, I would say 100 milliseconds maybe. You time to peak when the photon hits the rhodopsin. In amphibian, geckos, frog, toad, right, they live wherever the ambient temperature is. Maybe take seconds, 1 or 2 seconds. Yeah, but still, in terms of, you mentioned about time scale, 1 or 2 second time scale is still much, much faster than hours or if not days of G protein signaling, like a glucose level regulation by glucagon and epinephrine.
But if you're a gecko and you're looking to detect something, you could be a second behind the curve of what's happening in the world.
That's why you cannot be too big. They don't have enough to eat. Just kidding. Okay, you can edit this part out. I'm just thinking out of my hair.
But it's amazing that we're lucky that ours is fast. I mean, that's because I can't imagine being a second behind the visual world and still expecting to intercept catch a baseball or something like that.
Catch a baseball is actually I do a little calculation on that one. So assuming a baseball like 98 miles per hour is the limit, human limit, am I right? Yeah, so the reaction time, from the balls through the pitcher's hand, right, and reach the plate when the hitter need to hit it, is not enough for us phototransduction recovery to have any effect. So most of the speed coding and whether an outfielder can catch a flying ball and the hitter can hit the ball is probably pretty much not rest on phototransduction.
It's a prediction.
It's a coding. Somewhere in the circuit, the entire visual system had to factor that in within a few milliseconds. Yeah. So again, nobody say vision is solved, okay? But I just happen to be in the easiest part of the visual system, readily available at the experimental level to me, and make a living from there.
Okay, so there's two things I could worry about in time, thinking about it the way I am. So one is just sort of the latency. Something has happened in the world, like a flash of light. How quickly can I react to that? And then the second is, if two flashes of light come close to each other,
you don't distinguish them.
How close can they be? And I see them as two. So not only does the photoreceptor has to respond quickly, but it also has to recover from that quickly and be prepared to respond again. And so so what does that? What turns off what gets that current turned back on?
Right. So you asked more than two questions. So I will answer the one I know how to answer first. Because Mel Simon asked me the question before. Because in his lab, I studied photoreceptor recovery. But when we talk about how fast things may happen, like a hitter have to hit the ball, right? So that point, phototransduction, that even not reached the peak yet. Okay, so the coding, the calculation has nothing to do with photoreceptor recovery. But if you want to have the vision to discern, let's say, movement from point to point, right? Then you don't have to rely on point A's phototransduction being turned off. Because all you need to worry about is how fast does it move from this photoreceptor to the next, right? So the difference there could be microsecond, could be millisecond, but as long as the brain can tell them apart, no problem. Phototransduction can take as long to recover, but for the vision to work with less than 100 millisecond time resolution, still doable. Okay, so that answers your question. So that's why we have so many photoreceptors in the retina. So it's not just one photoreceptor do all the vision for us. It's actually they divide the labor, that kind of stuff. So second thing, how fast we lose visual temporal resolution. You all know that the fluorescent light, right, is driven by AC, right? Mm-hmm. So AC is here is 60 hertz. So actually we can recapitulate that when we record ERG from normal human. You can make flickering light, right? And at the fusion where you do not see any discernible peak, that's what we call a critical frequency fusion frequency. Is always less than 60. That's why, this thing is flickering, but we cannot tell it's flickering because our phototransduction cannot catch up. But if you were to use a fast camera, right, you know about this, and then you do it and you slow things down, of course this thing is flickering. It's just we cannot tell. So don't quote me on this one because no one asked me this question for a long time. I used to remember. I think a human cone flicker fusion frequency somewhere around 45 to 55 hertz. Yeah. And mouse is even less. Mouse probably 30 to 40.
So what molecular mechanism in the eye is responsible for that? Or is that a brain thing? Or is the eye not able to follow that high frequency flicker at some point? Yeah, you said the electroretinogram shows the flicker fusion.
55 hertz. That's pretty fast already.
That's pretty fast.
Yeah.
So that means that the photoreceptor recovery
doesn't matter.
The photoreceptor doesn't what matters for that?
Okay. Let me ask this. Let me use the one clinical case to answer your question. So I talk about RGS9, right? So I'm the first one actually make the human geneticists to follow my work to look for RGS9 mutation in human. They look very hard and they found several. So those guys, how do they find them? They have a cohort, this is done in Harvard. So they have a cohort of people that they take registry on, that they have some kind of family history, and they should have some kind of phenotype, but they haven't had one yet. Okay? So that means sit them down, and they just project dots flying across the computer screen. You can control the size of the dot. You can control how fast the dot fly across the screen, right? And that's a temporal resolution. Those guys have a defect seeing fast-moving things. So human RGS9 mutant is not the right term. Very disrespectful, I'm sorry. So people suffer from not having functional RGS9 in photoreceptor. They coin a special term for them called bradyopsia. They have normal daytime vision. They can live normal life, no problem. But they cannot play ping pong or tennis. They don't see the ball.
Great. That's exactly getting at the thing I was talking about. So let's talk about RGS9.
Yeah.
It sounds like we should understand what RGS9 is doing. Would you give us a little summary of RGS9?
Yeah, so RGS9 is in a protein complex called transducing gap, and G means GTPase, A is accelerating, P is protein. So it's a gap protein. That's the protein that help G protein hydrolyze in GTP. G protein is a self-timer. It binds GTP with the help of the activation factor like a activated G protein coupled receptor. And when it binds it, and the internal GTPase activity will hydrolyze GTP into GDP. And when that thing happens, eventually, and G protein alpha will reunite with beta gamma and go back to the inactive state, right? That's the cycle. RGS protein is discovered back in the turn of the century that we know very well for years that if you just grind up a bunch of cow eyes, which you can buy $1 per piece from Tennessee or Kansas, right? You grind it up and you follow the GDPS, you can isolate very pure transducing. And then you can throw in radioactive GTP gamma 32 label, right? And you just assay how fast G protein hydrolyzes the GTP. In that assay, the turnover time is 20 seconds. You close your eye, you don't see anything because the moment you close your eye, the G protein activated previously already deactivated. So we know something inside our photoreceptor help transducing turn off fast. And that's why we coined the term gap. Something has to activate transducing GTPase activity. What is that something? Right? This is the biochemist's perfect PhD project for biochemists. You know what you want to assay. Just fractionate the protein, sort it in, see which one accelerated, right? It's been done. And turn out that, if you throw in the PDE gamma, the effector, it will help transducing hydrolyzing it. But that experiment is tricky. If you isolate the PDE gamma from the eye, it will work. If you use recombinant PDE gamma, it will not work. So that means that maybe something you co-isolate with PDE gamma is actually the activator, stuff like that, okay? So then, lo and behold, in 1995, in the C. elegans field, I forgot what they studied, but anyway, some of the G protein, they have mutants. That mutant, they help one of the G protein alpha in worm hydrolyzing GTP. And then, that's before the Human Genome Project completion, right? So then when they published the work, right, and we in the eye field got so excited, this might be the missing link. At that point, we have no idea how big is this gene family, right? And as the worm people call them regulator of G protein signaling, RGS. So then that time I was a postdoc. Need a career defining project. And then turned out that my thesis advisor, he need a fundamental question. So a lot
perfect combination.
Except that you had to have competition with the lab who give you a PhD degree. So anyway, we go in degenerative cloning. I found the RGS-R. R means retina. Later on, they rename it to RGS-16. And then Jim Hurley found RGS, retina RGS. Okay, some other people find RGS4, Ted Wenzel in Baylor found RGS9. And then later on, and then we all express recombinant RGS throw into the test tube, and we all prove that they can help transducing hydrolyzing GTP. So very quickly, the field as the next question. Okay, so transducing hydrolyzed GTP through the help of all so many RGS protein, or just one of them is the real one? So how do you find that out, right? And this is perfect when gene targeting can answer this question for a lot of people. And then if you, so it could be redundancy built in the system that G protein has to hydrolyze GTP very fast, regardless who's the helper, okay? Or there's a dominant one. So at that time, I made a critical experiment. I told Mel Simon that, I don't know, I know the in situ hybridization in the lab hasn't been attempted, okay? But I want to do that. He says, you can go where to cut section, that kind of stuff, okay? Hybridization oven, just borrow those things and do it. So I just put all the candidates together. RGS-R, RGS-4, 9, and red RGS, right? I checked all them out. And then I'm looking for photoreceptor dominated RGS protein. It turned out to be RGS-9. So then, but that's not my protein. I didn't clone that one. So Ted Wenzel cloned it, right? So I called up Ted Wenzel. He's a very respected biochemist in the field. I said, Ted, if you don't mind, I want to knock out this gene. I don't want to stay in real dose. So if you are not onto it, and Mel and I, we are interested. So do I have your blessing? Ted Wenzel said, of course, let's collaborate. You guys make the animal, and if you don't mind, let us do the biochem, right? And we let Dennis Baylor and Marie Brent record from the animal. So actually, that's my career-defining finding. So I abandoned the gene I cloned, and instead I focus on RGS-9, and I knock it out, and I show the slide, right? So when you knock it out, the time to peak is almost identical. But you can see very clearly the recovery phase, right? So the recovery time constant moved from 200 milliseconds to 2 seconds.
So that makes perfect sense for the symptom that people experience when that isn't working, right?
Right. So then, but if vision work by ignoring the recovery of phototransduction, then how slow the photoreceptor recover shouldn't matter. But why would those people have this kind of bradyopsia issue?
Yeah.
That's an open question. Nobody studies this, I think. This has something to do with coding, how the photoreceptor signal being selectively interpreted by either retina itself or by the thalamus or by the brain to selectively extract whatever information they want to interpret. You see what I mean? But if you make things slow, okay, to a point that the interpreting center become confused, and they will show up in behavior, in your overall visual performance. Stuff like that.
So the rest of the retina can only extract out what information is there. If the information about time is completely smeared and lost, you can't fix that.
Well, that's my favorite. I mentioned about whether like ball players, like, they may have a more RGS-9 than us. So if the recovery is not important in time coding, in vision, right? But I also had a collaboration with a couple, Salacio in upstate New York. They are the first few to introduce OKR measurement in the eye field, in mouse, okay? So when they look at the animal that has faster raw phototransduction, slower raw phototransduction, what they find out using behavioral assay is that they do have a phenotype. And for the RGS-9 knockout, and then their ability to discern the moving parts, the speed gets slowed down. So at behavioral level, you slow down phototransduction, you do have a disadvantage, right? Which is explains the human phenotype. But in the RGS-9 overexpressor, you have a faster raw recovery. So the single unit response time frame is shortened. Do they confer any advantage in that behavioral assay? The answer is yes, conditionally. Condition in a way that in the purely scotopic condition, meaning that the light intensity only allow to activate rod, but not cones. Having a faster phototransduction in that assay do nothing. So if you do this in photopic condition, where the light is so strong, rod already saturated. Anything you see come from the cones And then the temporal resolution won't change, which makes sense because all the manipulation is in rod. Only when they use the mesopic condition, where light intensity is enough to activate cones, but not enough to saturate the rod. Mesopic condition, having a faster rod, confer better vision.
That's great. I mean, it seems like it almost would.
Are you following me?
Yeah,
yeah.
Great. It seems like it would almost have to be.
This is actually the first time I have this kind of in-depth discussion about my collaborators' finding. And then, so that means that, and rod and cone, later on, we know that they couple. They are gap junction couple. So maybe in the mesopic condition, because they are couple, right? It's like bring your kid to hiking something. You know, you want to go that high, right? But you're slowed down by your kid. But if your kid is, having a few energy bar, can catch up with you, they can go higher. That kind of analogy.
So cones are faster anyway, right?
Cones are faster, yeah, because they have a higher RGS9 concentration.
So if you're a cone and you're being coupled to a rod that's slow, that guy is slowing you down. He becomes the weakest link.
Yeah, it's the weakest link. So in a mesopic condition, but if you were to speed up rod, right, and then rod can somehow more keep up with cone, that confer better vision.
And then you go to brighter light where the rods aren't really contributing because they're all just saturated.
True.
Now it doesn't matter.
Right, it doesn't matter. Yes.
That's great. I mean, that's.
I'm glad that you catch on immediately.
So maybe, how can we all get our rods sped up? Maybe I could be a...
No. Actually, what I really want to push the story is actually try to get the primate people to allow me to elevate the RGS9 concentration in their monkey, for example, because they have fovea, cone-rich area, right? I think there's a gene delivery tool that you can target fovea very specifically. And out of this protein complex, I know how to overexpress which component to bring up higher concentration in the gap, transducing gap. So if I do that in primate, we can do flicker fusion ERG recording or some behavior recording or even perimetry study, right? Look at temporal resolution. If that's the case, that if I elevate RGS9, they have a faster cone phototransduction. Now we have a super monkey. I can see better. Stuff like that. Yeah. Those are doable experiments.
They're genetic manipulation or?
Just virus. You just deliver virus. Virus that will target the fovea,
Yeah.
Stuff
so there are people whose RGS9 is not fully working. Are there people who have more than the normal amount of it, or
I don't know.
Version of it?
I don't know. No one volunteered to let me get a piece of the retina and measure. But this kind of stuff, I don't know. I really don't know. What I like about it is that the idea of RGS9 level, we are not talking about a huge change. Twofold change can bring up significant speed up of a vision, right? And this gotta be explored more to see whether there's any, therapeutic value or some competitive value, in sports, for example. Yeah.
Does it matter whether it's whether it's I'm just thinking, for example, in development, right? You talked about AMD, which is affecting more the older people, right? But let's say when you're more of a young person, when you have fully functioning vision, would the advantage be more for one group of age group versus the other? Have you looked at that in mice or not yet?
I don't think anyone actually. This kind of project is very difficult to get funded. If you listen to me enough, I can inspire you. But if I want to just our past conversation, put it down in 12 pages, right? Get reviewer to get really excited and want to support the grant. That will be very, very difficult.
That's what everybody says about what they do too. That's just the way it is. Yeah, it's not the phototransduction isn't the last G protein step in vision either, is it?
It's not.
There are more.
There are more. And then we know a lot more than so photoreceptor gives the signal to the bipolar cell. Bipolar cell in turn relays the signal to ganglion cell. And at the junction of photoreceptor and bipolar cell, we have interneurons called horizontal cell, and they will regulate how the glutamate-based information transfer being regulated. And then between bipolar cell and ganglion cell, there are a group of interneuron called endocrine cell without axons, right? They also regulate the transmission. So retina is wired to be able to compute by exactly how they compute, what they compute, and who is doing the computing for what stimulus is still a very, very early stage of being mapped out. Yeah.
And some of the communication between neurons is by G protein coupled receptors, and some of it is not G protein coupled receptors. Is that right?
Yes. So in bipolar cell, the on response in depolarizing bipolar cell, that G protein receptor is already known. It's a metabotropic glutamate receptor number 6, mGlu6. The G protein in that pathway is G-alpha O. Okay, O stands for other. When they in the old day when they isolate the G protein, right, and they find out, whoa, there's a very abundant G protein in the brain. That other G protein is just O. And so it's not olfaction. It's not olfaction. Olfaction, O is G off, OLF. They spell it out to distinguish. Okay, so G alpha O, and then through unsettled mechanism will lead to the closure of the channel. The channel is TRPM1. So when that channel close, okay, and then the photoreceptor bipolar cell will hyperpolarize. Okay, but because the first vision synapse signal transmission is opposite. So when photoreceptor get activated, it hyperpolarize. So then the mGlu6 in the dark is constantly bombarded with the glutamate dumped out by photoreceptor. Now photoreceptor stop to dump out glutamate. mGlu6 fail to activate GO, fail to inhibit TRPM1. TRPM1 open, and the bipolar cell depolarize.
So is that also very fast?
Yeah. Close your eyes. What do you see? Nothing, right? So that means that whatever TRPM1 already be shut down, right? By the time I want to get this right, but it's very fast, millisecond.
So but the other set of bipolars are getting ionotropic?
Yes, and those are called off type.
So we normally think of ionotropic as being a lot faster than metabotropic, and is that not true in this case?
That's beyond my expertise because first of all, I'm not familiar with GluRs, okay, and if you really put my feet to the fire, I would say I understand metabotropic better than ionotropic. Okay, yeah, it is true because you would think that being a ionotropic one, right? You bind glutamate, you open, right? It's just or yourself, right?
There's 2 steps.
Yeah, right. As opposed to you have to have a protein interaction, you always have to factor in diffusion. Mm-hmm. If they are not in the same place to begin with, right? And diffusion takes time, interaction takes time, conformation change takes time. GluR 2 steps. Binding, conformation change, open. Yeah. So, but
I was just worried that on and off would be kind of out of sync with each other because of the speed difference, but apparently it doesn't affect anything.
Well, I think brain is smart enough to figure out the difference.
That out.
That's the best hand waving answer I can give.
Hey, Jason, thank you very much.
Thank you so much.
It was really fun. It was very lively. Well, we've used our time. If you want to.
No. Oh my God, good time go by fast, huh? Thank you so much for having me. I really enjoyed the visit.
Yeah, me too.
Yeah.
And thank you.
Thank you very much.
Cool. This has been Neuroscientists Talk Shop.